If I had to come down to like what are the two main reasons why people age, I'm going to say it's because we eat and we breathe. We're studying this in Alzheimer's disease right now. And so basically what happens is immune cells in the intestine can also end up in other tissues. At least with mice right now, we've identified immune cells that recognize bacteria living in your gut. These immune cells are also present inside the brain. One of the things that stress does is it dampens your immune system. And so that would allow, virus reactivating. These things happen when your body's under stress, right?
It's rare for scientists publishing in peer-reviewed journals to also go all the way to a shipped product. What was that journey like?
So going from an idea to a product that could potentially help people, it's not an easy journey. Usually what this starts with,
Dr. Dan Winer, welcome to the Augmented Life. I'd like to kick things off, I think, with a question aimed at you, Dr. Winer. Your earliest work really showed how adaptive immune cells homed in on multiple systems, right? Adipose tissue, liver, gut, during obesity, driving insulin resistance, implicating, I think, immune system adaptation to metabolic disorders. This blew my mind as I was doing background research before having you on. What led you in this direction? How did you set off in pursuit of this discovery?
First, thanks for having me on.
My pleasure.
I got interested in studying, obesity and metabolic disease. This was kind of something that spun out of, conversations I was having with my twin brother. So I have a twin brother who also does research. We used to, keep in touch. Obviously we didn't live in the same place. By the time, I started going out to do my postdoc, I was in California. Sean was in Toronto. So one of the ways we used to communicate with each other, and this is kind of nuts. We used to play computer games, and online we would talk about, ideas of how, the immune system could potentially modulate many diseases.
During like voice chat over video games?
Oh yeah, that's what we used to do. One of the things this was probably back in 2006, 2007, around then. One of the things we were interested in is the adaptive immune system, which are T cells and B cells. So there's two broad types of immune systems. There's the innate immune system, which, recognizes, distinct broad patterns across, invaders like bacteria, and senses danger. And then there's also the adaptive immune system. This is the immune cells called T cells and B cells. These basically recognize very distinct kind of laser focused patterns, in bacteria, very specific. And we were wondering, at the time, these immune cells, T cells and B cells, they weren't really, linked to diseases outside of autoimmune diseases, type 1 diabetes, multiple sclerosis, rheumatoid arthritis. And at the time we were talking about, what do these T cells and B cells do to other diseases, including all these chronic diseases of aging, cardiovascular disease, neurodegenerative disease, metabolic disease like obesity and diabetes. And that was, my brother's lab was studying type 1 diabetes. And so this was a natural model at the time to look into type 2 diabetes. And so, we've done some research. There were some studies coming out, very early on T and B cells in the brain, but the adipose tissue, which is the fat, was the tissue that was most affected in obesity and diabetes. And so that was the kind of the tissue we started looking in. And that tissue then showed profound changes in T cells, in B cells and adipose tissue. At that time, so that paper, we published two papers, and it was 2009 and 11, showing that T cells and B cells can modulate glucose, and also, obesity. There was a couple other papers that were published around the same time showing the same thing. And then we moved to the gut from there because, the adipose tissue we thought was kind of like, an organ downstream of where this is all starting. And we thought, what drives this inflammation and fat? And inflammation and fat is in part controlled by these T cells, B cells, and other immune cells, including macrophages. But what controls this? And so that's when we thought about the gut. So the gut, we think, is upstream of the fat. it's actually the first contact you have, with so many different varieties of, antigens. And so this could be the bacteria living in the gut, the food you eat, all of these things interface with the gut. And then, of course, the largest immune system in the body is also living in the gut. And so, it was this interaction between the gut immune system, the bacteria in there, and the food you eat, which, sets the inflammatory tone in the intestine that modulates the flow of, these products, from the bacteria downstream to get into fat. And that's how we got into the gut. And so, I mean, this was like a gut to fat axis, but the gut also to the liver. And so we started looking in the liver as well. Now it's been showing the gut connects to the brain. We have a paper, a preprint out showing how the gut connects to Alzheimer's disease through the immune system that's in there. And so, yeah, and that's basically, how all this started. It started with me and my brother talking over a computer game about, what else can this adaptive immune system do? I mean, it's got to do more than just autoimmune disease. And this was, way back in 2006, 2007.
Wow. Yeah. So I saw in those early papers you were talking, and I think maybe even you coined this term of immunometabolism, and that has been work that's been built upon. I've since then seen this term metaflammation, trying to put a finger on the uh, inflammatory effects of metabolic disorder, or vice versa. I guess the causality runs in both ways. What is there a general lesson to be learned here about not just metabolic disorders, but also aging and senescence?
Yeah, okay. Yeah, thanks for the question. I mean, we never actually coined the term immunometabolism. It was a paper that came out around, I think it was 2010 or 11. But some of the studies we did were kind of pillars in the field, showing that the adaptive immune system, which is basically like half of all of immunology, contributes to, metabolic disease, and that wasn't really appreciated before. But the term was coined by another group. So meta-inflammation is this kind of, chronic low-grade inflammation. I think it was initially used to describe inflammation with metabolic syndrome. It's typically, and this is, of course, is different from acute inflammation. Acute inflammation is, what you typically think about when something gets inflamed. Like, so say you cut your finger, it gets red, swollen and painful. These are kind of, manifestations of acute inflammation. Meta-inflammation is typically this, low-grade chronic inflammation. As it said, it was initially described mostly with obesity. It's typically thought to be more of a sterile kind of inflammation, but whether that's truly sterile or not, I'm not sure, because I think some of the inciting agents which drive meta-inflammation are probably linked to, either bacteria, bacterial products, potentially viral reactivation. And so I don't think it's truly a sterile inflammation. Like, and then you asked about, how does this, contribute to aging, and, what is it just, inflammation, and aging is quite interesting. So that's my lab studies that, a lot right now. We're at the Buck Institute for research on aging. And basically, when people ask about aging, like whether it's, due to the environment, or whether it's purely genetic. And most studies coming out saying that aging is probably driven, at least I'm going to say 80% by the environment, and maybe only 20% or less by genes. And the environment could include, many things like diet. So diet is, I started out, as an obesity researcher, but diet, stress, the way you sleep, radiation in the atmosphere. I mean, there's radon gas, for example, in areas, pollutants, chemicals. These would even be things like, plastics, physical inactivity, um, and chronic infections. Like people have, latent viruses that they're constantly kind of, fighting off. Cytomegalovirus is kind of one example, and these are critical things in the environment. But if I had to come down to like what are the two main reasons why people age, I'm going to say it's because we eat and we breathe. And so, I mean, and you can't do too much about that. I mean, you can eat less, but when you breathe, I mean, a small amount of the oxygen that you breathe in is actually ends up as reactive oxygen species produced by mitochondria in your body, and this damages our cells, and this causes gradual wear and tear over time. Eating is basically, whenever you eat, your body makes insulin. Insulin is a hormone that builds things up, and that's called anabolism. And then whenever you build stuff up in your cells, they're stronger, they can divide. There's chances for errors in there. Every time your cell divides, there's chance that the DNA can get damaged or altered. Um, and this DNA damage is part of what's called the hallmarks of aging. There's like 12 of them. And there's a whole bunch of them, like, your body's ability to recycle things. Some cells form these quote unquote zombie cells, which are senescent cells, where they stop dividing permanently. Some proteins get damaged. The microbiome changes. We'll talk a little bit about that later, I think. And chronic inflammation sits in there as well. So chronic inflammation is just kind of one of maybe 12 different pillars which define, hallmarks of aging.
Yeah, gotcha. And no matter what, I mean, the simple fact of the matter is that, as you point out, oxidation is named after oxygen. It's a good thing, and it's also a bad thing. Well, I'd like to drill more into the insight about digestion, about everything that's going on in the gut as being upstream of metabolic disorders and literally being upstream of obesity. And you pointed out the role, of course, of bacteria that live in your gut, the microbiome. I think every one of our listeners has heard about the gut microbiome and has some understanding that some elements of the microbiome might be helpful, some might be unhelpful, that you can regulate these populations with probiotics and prebiotics. But the actual, let's say, mechanisms of action, the ways in which these things can be useful or not, aren't really, uh, the arising from the microbiota themselves, but rather what they produce, right? The postbiotics. To talk to us about what postbiotics are, and in particular the ways in which you've been exploring their medical effects.
There's prebiotics, there's also like probiotics, and then there's postbiotics. So there's a lot of these different biotics. So a prebiotic is something that's upstream of the bacteria. So in the gut, for example, or anywhere. I mean, in theory, I mean, although it's not common, I suppose there could be, people could ultimately, do something with lung or skin, for example. There's microbiota there, but the most common one is studied is the gut. So a prebiotic would be something that the gut bacteria can take in, eat up, potentially, metabolize and turn into something else. So the probiotic would be, giving the bacteria themselves, and then the postbiotic is what the bacteria actually produces. And so, these things would be something like the most common studied things would be metabolites. A common type of metabolite would be short chain fatty acids. So these are small fats that are produced from soluble fibers usually. So the bacteria picks up these soluble fibers and digests them and turns them into these little fatty acids. Some of them are called, like acetate, butyrate, propionate. These would be kind of some of the main ones that they produce. These postbiotics have profound effects on the cells of the body. So in the intestine, for example, if a bacteria nearby starts making some of these short chain fatty acids, these things are actually used as fuel sources for the cells lining the intestine. So it's like a symbiotic relationship. The bacteria can use these prebiotics to make, short chain fatty acids, which potentially some of them could use as fuel, but our own body uses it as fuel as well, right? And so both organisms need. But the other thing that these these postbiotics do is that they also have a very important immune modulatory effects. So these things can basically, be used by the immune system, and alter, the types of, proteins that they make. One of the most common described phenomena was, these short chain fatty acids acting on on T cells and or regulatory T cells, and boosting functions of regulatory T cells, so that these cells then can produce, cytokines which dampen inflammation. So this one example would be interleukin 10. Um, and so or Yeah, so basically, like, it's a symbiotic relationship. The bacteria use these, prebiotics, and then they make postbiotics, and then postbiotics can be used by both bacteria and your own body. So, and it's not just metabolites. They can produce proteins or maybe like enzymes or peptides. But the most common study thing, and what we study linked to Propion, are the metabolites that these bacteria produce. So these would be the postbiotics of interest for us.
Yes. Wow. All right. So what led you to the propion molecules?
So my lab has always been interested in, in studying, as I mentioned, that, as you talked about before, the gut immune system. So how does the gut immune system, regulate diseases from the intestine, and diseases that are, elsewhere in the body. So, there's a lot of people studying, things like inflammatory bowel disease. I mean, this is basically, the gut becomes inflamed, the immune system is inflamed, is locally in the gut. My lab is interested in how the immune system from the gut controls diseases elsewhere. So for example, how the gut would control the brain and Alzheimer's, or how the gut would control type 2 diabetes or insulin resistance from the fat, or to the liver. And I bring this up because some of the receptors that these immune cells in the gut express, one of them is called the aryl hydrocarbon receptor. And so this was a molecule that we were interested, and we published a paper back in 2019 showing that you can intervene by targeting the aryl hydrocarbon receptor in these immune cells in the intestine, with a herb supplement called Indigo naturalis. And so we published this paper in 2019. This herb basically binds weakly to the aryl hydrocarbon receptor and dampens inflammation in the gut. That then in turn strengthens the intestinal barrier, and and improves the relationship between the immune system and the bacteria living in the gut. So that was in 2019. We were interested in this AHR ligand. And then, it was a couple of years ago how I got interested in these propion molecules. This was actually Nicholas approaching me. And, while he approached me on to look at molecules that were, intervening or binding to the same axis we were already studying, it was his idea, and he basically came up to me. And he was an undergrad student at the time at UC Berkeley, studying neuroscience, and asked me if we were interested in studying some specific indole metabolites. He showed me a paper by a person, a PI in Germany. It was Burkhard Pogler, a paper about one of these indole metabolites, vastly improving lifespan in a primitive rotifer model, and asked me if I was interested in studying these in longevity. And I said, yeah, we're not just interested in longevity, we're interested in, all possible diseases and uses that these molecules could be used for. And that's kind of how Propion started. And I'm not sure if Nicholas wants to add to the story about that. I mean, he approached me. I'll be the first person to admit it when somebody comes to me with an idea. I kind of just wanted to add a little bit on how Propion links to some of the research my lab is doing right now as well. So my lab basically has been dissecting what happens during diet-induced obesity and aging at single cell resolution in the immune systems that live in the intestine. And so one of the things we're finding is if you look at cells on a cell by cell basis of all the different types of immune cells that live in the intestine during diet-induced obesity, some of the cells that are most impacted are things called innate lymphoid cells. So T cells, B cells are impacted, of course, but innate lymphoid cells, which is another type of immune system, these are immune cells that kind of look a little bit like T or B cells, but they have less specificity and they're kind of more broadly reactive to danger signals in the environment. Um, and basically, what we're finding is that a lot of the receptors that these cells express are modulated by some of the potentially indole metabolites, and so and that's also especially so we're finding some specific types of innate lymphoid cells that express the transcription factor called ROR gamma T. And these types of innate lymphoid type 3 cells, for example, are actually reduced in obesity and in diet-induced obesity, at least in preclinical models. And these are the cells that are very sensitive to taking downstream or tryptophan metabolites or like indoles. And so yeah, that's just linking in how metabolites that are produced related to propion could impact a whole variety of different types of immune cells living in the intestine.
Yeah, I gotcha. All right. Now, for listeners who haven't perhaps memorized a massive taxonomy of ligands, I think I should point out that the indole metabolites that we're talking about are categorically linked with the propion molecules that we're talking about, which are specifically indole 3 propionic acid and indole propionamide. And those are themselves somehow tryptophan derived postbiotics, though even that linkage is actually unclear in my mind. So, can you unpack that for us?
Yeah, so the way this tryptophan metabolism works is, basically you take tryptophan in your diet, and then tryptophan is degraded by different types of bacteria that live there. And so you've got, some bacteria like lactobacillus, for example, they can metabolize tryptophan into this postbiotic called indole 3 aldehyde. Another type of bacteria called Clostridium sporhyngeus can metabolize tryptophan into something called indole propionic acid, which is kind of one of the molecules we're interested in. But we're also interested in indole 3 aldehyde. Other bacteria can break down tryptophan and form directly something called indole. Indole is like the backbone structure of the molecule, and then when you add something else to it, if you add it on the 3 position and it's an aldehyde group, it's called indole 3 aldehyde. If you add a propionic acid, it's also in the 3 position, I think. It's called indole 3 propionic acid. So these are the molecules that tryptophan is metabolized into. Actually, most of tryptophan is actually taken up by the host. So I think it's like probably more than like 90% of tryptophan is metabolized by enzymes called IDO and TDO. And so these basically, I think TDO probably, I think it turns tryptophan into a precursor for NAD that your body can utilize as energy. But these pathways also can produce potentially inflammatory and more hazardous molecules. And so the goal with propion or is to try to divert tryptophan metabolism away from the host and divert it to bacteria in your gut so they can start producing these beneficial indole metabolites, which are generally produced in small amount because most of the metabolism is done by the host. And so, yeah, and that's basically, how we want to use tryptophan metabolism. Some of it as well. Some of the metabolism of the host is beneficial, like in terms of antioxidant function. Like some of it can be converted to melatonin, for example, or serotonin.
And serotonin, yeah, exactly. These are it's tryptophan's an important precursor of both of those, I believe. So you got to give your body some.
That's right. So yeah, but I mean, a lot of the stuff that's that's from IDO and TDO, um, which is most of what you used. I mean, our goal is if we can divert even a small amount to help your body produce, maybe more serotonin, melatonin, IP, indole propionic acid, indole 3-aldehyde. That you should get some, really nice, benefits from these metabolites.
Right. That's astonishing. And going back to an observation you were making earlier, it sounds like there are beneficial effects not just against the mechanisms of inflammation that we were talking about at the start, but also like uh weird problems like leaky gut, if I was connecting the dots from what you were saying correctly.
Yeah, basically these metabolites, these indole metabolites, they have a number of beneficial effects. One of them is the intestinal barrier. So the intestinal barrier is important. It's basically, it's made up by a couple of things. The cells that line the intestine is one of the components of the intestinal barrier. But it's also the antimicrobial peptides that these cells make. So these and also the mucin that the cell that's produced by goblet cells lining the intestine. And so there's a whole bunch of things that compose the barrier, and even some of the antibodies you produce. In general, when the intestine is in a healthy state, or when you're feeding it a healthy diet full of dietary fiber and other healthy foods, what happens is that you've got abundant mucin in there. The amount of mucin that's in there is also regulated by the immune system, and so generally, if you've got you know, uh, cells that dampen inflammation, like regulatory T cells, for example. They can produce factors that drive mucin production in the intestine. Some other these innate lymphoid cells, including some of the innate lymphoid cells, type 2, can produce things that drive mucin. Almost like if you think about when you get like a runny nose, right? When you have an allergy, it's the same kind of immune system that drives, mucin production to cause your nose to run. Also, some of these cells, to a lesser extent, this also happens in the intestine, but the mucin is quite, protective there. Um, Also, some of the antimicrobial peptides that are produced in there that helps keep the bad bacteria at bay in the intestine. Those are produced by immune cells that also have these things called aryl hydrocarbon receptors. And so, these were some of the things that we were studying initially. One of the molecules that these immune cells produce is called interleukin 22 or IL-22. This causes cells in the lining of the intestine to make antimicrobial peptides. There's also some cells called Paneth cells down there, and they produce these things as well. Um, and so basically, it's like the immune system boosting the barrier. And why is that important? It's important because when your barrier is intact, it keeps this nice relationship with the gut bacteria, so they don't kind of get into your circulation, and they don't get deposit into tissues. Um, and some of the toxins and things they produce are kept farther at bay as well. Some of these toxins, one of them that's commonly studied is called LPS, lipopolysaccharide. That is thought to get into tissues, like including liver and fat, and act as an adjuvant to fuel inflammation for the immune system that's in there. And so basically when your gut barrier is not intact, more of these toxins get in and they act as adjuvants to fuel immune system. And the immune system, causes chronic inflammation in these tissues. When it happens with like the liver or the fat, this chronic inflammation, I mean, this is going back to the very beginning about metabolic syndrome and obesity and insulin resistance. This chronic inflammation in these tissues is thought to jam up signaling of the insulin receptor. And so, usually the insulin receptor likes to have one of its, molecules have this, tyrosine phosphorylation. It gets, phosphorylated in one area. But if there's a lot of inflammation, you get this different type of phosphorylation downstream, and that jams up signaling. And then when insulin doesn't work properly in fat, for example, the fatty tissues can't, adequately turn free fatty acids into triglycerides well. And so basically these free fatty acids, they start leaking out of the fat and then they deposit into other tissues. If you get too much free fatty acids going to the liver, you end up getting fatty liver disease. And that's a major, global issue right now. If you get these free fatty acids going into the muscle, they can interfere with muscle metabolism and strength. And so these are like all complications downstream of insulin resistance, metabolic syndrome, all starting at the gut.
Right.
Another thing I just want to mention, sorry, you're getting me because I like talking about this stuff.
I
Another
like
thing,
hearing about it.
yeah, another thing about the gut that we've been studying now, that we've published and or helped or contributed to papers on, is showing how immune cells from the intestine, they don't just sit there and function on their own, right locally at the interface. What they do is they migrate through the body as well. So, I mean, this happens, whenever you eat something, but also happens in the context of disease as well. So typically, whenever you eat something, there's like, immune cells, which are like, macrophage-like cells. They kind of grab bits of food and then they, pass them on to another type of immune cell called a dendritic cell. And then these cells can basically like migrate to lymph nodes nearby, and then these, that talks to T cells and B cells in the lymph nodes, and then they circulate, throughout the body, but they also end up making their way back to the gut. During disease, what we find, and this was a paper, we contributed to with, multiple sclerosis, but we're studying this in Alzheimer's disease right now. It's part of a preprint that we had. And so basically what happens is immune cells in the intestine can also end up in other tissues. I mean, in MS, I think it was in the lung, but in all, they can also end up in the brain. And so at least with mice right now, we've identified immune cells that recognize bacteria living in your gut, being these immune cells are also present inside the brain, or at least in the meninges of the brain. And so we think these immune cells start out in the intestine, and if the relationship there with the bacteria isn't good, or the bacteria changes in there in a bad way, that's called dysbiosis. That can contribute also to inflammation and, modulation in other tissues like the brain as well.
I see. All right. So when this goes really bad, you see big name disorders like IBS, like Crohn's. But it sounds like there's actually a spectrum, and on a smaller scale, you could just trigger inflammation across multiple systems.
That's right. And so even if you think about diseases like Crohn's disease, so Crohn's disease has a number of like extra intestinal manifestations. And so even though the disease is most, commonly, centralized in the intestine, it's actually a systemic disease, which means that it impacts multiple, organs and tissues in the body. And so we think similar things are happening, not just with Crohn's. We think most diseases, I mean, have a component that's probably related in some way or another, to your immune system reacting against either bacteria or viruses. And some of these are probably found in the intestine to start. I mean, in some cases, I always tell people that, most autoimmune diseases, I mean, people don't really know what they're caused by. And I'm saying most of them are probably going to end up being caused by, a virus or a bacteria. Whether those start in the intestine or whether they start elsewhere. I think even you saw like multiple sclerosis is caused, in part by an Epstein-Barr virus infection. I saw another paper that just came out recently showing that a disease in the liver, I think was primary sclerosing cholangitis, which they bile trees in ducts and the liver gets scarred down. No one had any idea what caused it. There was a paper that just came out showing that this may also be caused by Epstein-Barr virus. And so, yeah, so there's been a number of papers on type 1 diabetes showing that potentially there's immune system cross-reacting onto, bacteria living in the intestine or other types of bacteria, living in the intestine, which has similar patterns to your own self proteins in the pancreas. And so that cross reaction, also known as antigenic mimicry, leads to your immune system crossing over to your own tissues. Uh, And so I think a lot of diseases have this. I mean, not all of them start in the gut, but I think a lot of these chronic diseases of aging, like neurodegenerative diseases, and potentially even someone in my lab is also Taylor Valentino. He's also studying sarcopenia right now, how that potentially could be modulated by bacteria in the intestine. Tumor immunotherapy, bacteria in the intestine. There's a lot of things. So the connections, are abound.
Yeah. Wow. All right. Well, this is both astonishing and perhaps exciting. I, as a complex systems thinker, I could see how a wide variety of autoimmune disorders are runaway feedback loops that have gone, in a dysregulated sort of way. But it would make sense that there's some trigger, and and it could just be that the trigger is undiscovered, but ultimately is a viral or bacterial trigger that kind of like begins the cascade.
Yeah, I mean, if that's true. If you think about it, a lot of the triggers, I mean, say we go back to Crohn's disease, is stress, right? So a lot of people, they get a Crohn's disease flare when they're under stress. And so why is stress so important? I mean, one of the things that stress does is it dampens your immune system. And so that would allow, if it's a bacteria, some people think of Crohn's disease might be linked to a type of mycobacteria, which is a different type of bacteria, but in other diseases, maybe if it's MS, it could be a virus reactivating. These things happen when your body's under stress, right? So it's not surprising that stress, which dampens your immune system and would allow bacteria or viruses to flourish, would be a potential trigger that happens during these processes.
Yes. Okay. All right. And this is another way of implicating the gut in a wide variety of, let's call it, defensive operations, because we're mostly covered with some pretty good dead epidermis protecting us from the world, but we have to eat. We have to incorporate a whole bunch of unsterile stuff, and stick it into our bodies, and so the battle begins there, and therefore the ability to fortify the defensive barrier begins there as well. Um, which, yeah, so that brings us back to the your indole investigations. Next, I want to just understand the journey from scientific discovery, the kind of insights that we've just been talking about, to actually getting a product in people's hands that can improve their lives. It's rare for scientists publishing in peer-reviewed journals to also go all the way to a shipped product. What was that journey like?
Yeah, so going from an idea to a product that could potentially help people, I mean, it's not an easy journey. Usually what this starts with is you have to, first of all, we had to decide on our product. I mean, whether we want to develop a prebiotic, a postbiotic, or a probiotic, which captures, these bacteria, or what they produce. Um, we had a number of considerations, what we wanted to do. In terms of, initially we're like, maybe we should just try a probiotic, and give the bacteria themselves. However, if we look across the literature on probiotics, the use is kind of variable. Some work better than others. There's a lot of, there's more regulations, this type of strains that are in there. It's a lot of detail. If you look at, uh, you know, postbiotics, giving metabolites themselves, um, that's also a very promising approach. However, there could be some issues with giving large amounts of um, postbiotic, especially ones that been have not been extensively reviewed in clinical trials, for example. So we'd have to set up more trials if we use postbiotics. And then there's prebiotics, which we could use as something from materials that are generally recognized as safe. Prebiotics I've always liked for a number of reasons, and this is why I think we went a prebiotic route in terms of developing our first product. Prebiotics have a lot of advantages because what they do is you take foods that you would normally eat that are safe in in pretty good quantities too, and they just basically feed the bacteria in your body. And so what they do is they feed the bacteria. So in this case, that we'd be feeding with Propion, we'd be feeding some of these Clostridia and some of the lactobacilli in there. The metabolites that the precursors that they need to produce beneficial molecules. And you're not really even limiting yourself to one molecule because you're giving them a nice prebiotic mix. They have the ability to produce many different types of molecules that are beneficial. So they'll produce, indole propionic acid. They'll produce indole 3 aldehyde as examples. They'll produce a number of things. I mean, they may even, produce, a little more serotonin or melatonin maybe as well too. Not the bacteria, your body that is would do that. Um, but yeah, so that's for me was one advantage. I mean, of a prebiotic, and it keeps the gut bacteria that they're healthy because you're feeding it the stuff they want, so you can expand good types of bacteria down there. Now, if you give something a metabolite directly, and this is also a good approach. However, there's a little bit more uncertainty or unknown to in what that could do. Now, giving metabolites themselves have been used in a number of conditions. So, short chain fatty acids, I mentioned, for example, butyrate. So, you can give this orally, intravenously. You might not know, but short chain fatty acids like butyrate are also given to people who've had surgery in their colon. So, if people have had their colon removed and you're left with a stump in there, and so you have a blunt-ended colon as it's waiting to heal, that area gets kind of inflamed. And so people are given enemas of these short chain fatty acids or butyrate to help dampen inflammation. And so you can give, metabolites that way as well. Um, Now, there's other ways to give bacteria as well. There's a whole emerging literature, of course, of giving the bacteria through like fecal microbial transfer, and there's a lot of stuff going on with with infections like C. diff is one infection, and um, but the prebiotic was very attractive to us because we could get this product out sooner. Um, we could expand different types of bacteria, and we could do this all in a way that's considered safe for based on the ingredients that we use. And so that was our first product. And so it's a lot of work because you had to go from after deciding on the product, and then there was a lot of trying to get funding and pitching, uh, building up teams, for example. And Nicholas played a major role in doing most of this team building and also pitching. And from there, it's basically, you have to get the product manufactured, the labels all printed out. We have to decide what's in there, and this is stuff that's done mostly by the CEO. I mean, Nicholas was doing most of this stuff, I'd say. Um, and yeah, and then once the product's out, I mean, we just launched a few months ago. We've got it. I've started taking the product myself. My wife takes it. Um, so far so good. I mean, I feel great on it. Um, there has been so this product. I mean, there was an earlier, very preliminary study that was done in Europe on the baseline of these ingredients, and it showed a number of beneficial effects. And so I'd say, probably the next step is we want to validate some of these claims that were made around kind of the backbone of this product in terms of, how well it helps, things like sleep, cognition, how your body's aging, things like that. And so that's probably some of the next steps we want to do. But it's a long journey to go from idea to product, and it never ends basically. Because I mean, at this point, we still want to refine the product too, right? We want to keep. This is like version 1.0, and we want to make it even better. I mean, we've got a lot of excitement and potential with this product, and we think it works. However, we need to validate some of these things, and also we want to make it even better as we learn more about it.
That attitude, honestly, that is principally, I think, scientifically driven is what attracted me immediately. Like I I don't think I've told you this. After I heard you speak about your research, I well, while you were talking, I took notes and then did some Google searches on the notes, found the Propion website and like checked out later that evening. And I am not usually an early adopter of these kinds of things, but it was so clear that you were not just like repackaging someone's supplements or something like that. You had really leading a science-driven process, and that honestly immediately earned my trust. And then I learned, oh, this is exactly where you're headed next. You want to get this into more people's hands for a kind of, you know, citizen science investigation. Let's pool our observations and find out what's next and what version 2.0 should be like.
Yeah, so we're basically, I mean, the thing I would recommend most for people if they're interested to go to the website www.propion.com. There's a lot of information about the product that we have, the science behind it, some of the studies, both human correlative studies and also preclinical studies that were done in mice. We have an offer going on now where if people want to track their own data, and Nicholas can help answer questions on this as well if people are interested. People want to track their own metrics. We can send a bottle of the which is I believe it's I'm going to say it's a 2 month supply, or maybe it's a 1 month supply. A bottle of the Propion blend for free, so they can try it out if they track their metrics. And some of the things we're interested in is, we want people to track their biological age. Some, if they can, some basic sleep metrics, for example. I mean, if they've got a ring or a watch that can track sleep metrics. Ultimately, some things with blood, like their blood sugar levels, hemoglobin A1C, for example, and some basic readouts, for example, like liver function tests, kidney function. Also interested in things like blood pressure as well. But yeah, most of this information you can find on the website, especially how the shake works. And in terms of how we want to make it better, it's going to be constantly using scientific evidence based on data that we receive. One of the things we're thinking about doing, I mean, when we initially made the product, we took out some of the fiber with it because we wanted people with digestive issues or inflammatory bowel disease to be able to handle it better. Um, and I mean, if you've got too much, say, soluble fiber in there, for example, it can cause bloating and issues with people with irritable bowel or inflammatory bowel disease. Um, and so potentially we might, but fiber is one of the things that can help the production of these indole metabolites even better. So one thing I would recommend, if you can handle fiber, you could take the shake and add your own fiber, like inulin fiber to it, for example. You'll get an even stronger effect from the shake. We may be making a product where we combine them in the future.
Inulin's pretty easy to source but on your own, so I think that was a good call. I'm also like deeply interested in driving this kind of wide scale citizen science. So my company Fulcra, we also want to like help make this investigation possible just by helping people who want to participate to track their their vitals while they're trying the Propion blend and then report back into a pool so we can see what there is to learn.
Oh yeah, that would be incredible. And I mean, and I said that all this data is all going to be used to refine and further improve the product for as well. And I mean, ultimately, I mean, like, I think you're interested in where we go in the future as well. I mean, yeah, down the road, I mean, like, we're the company's interested in doing a couple of things. I mean, we're interested in obviously improving the blend that we have. Or we're also, still interested in also administering the postbiotic, directly. So taking some of the metabolites themselves and giving it in different formulations, uh, modifying these formulations or the the um, the molecules themselves to make them better. These are things we're also interested potentially in applying these molecules in different ways, maybe as skin creams, maybe as pet foods, um, things like that. So potentially maybe even using it for different different combinations or in clinical trials in the future. I mean, so for example, like the shake, for example, has a good amount of protein in it, and so potentially we're thinking about using this for things that link to aging, like muscle wasting, also called known as sarcopenia. Um, Maybe it could be used in combination with people who are obese and they're already taking maybe GLP-1 agonists. Our shake could potentially boost some GLP-1 production. So potentially you could take the shake with a GLP-1 agonist and then maybe take a lower dose of the GLP-1 agonist and supplement it with the shake. I mean, you'd have extra advantages here because the molecules that have been produced by this shake can also potentially improve muscle mass and dampen inflammation in muscle. And so, people who are taking a good dose of GLP-1, some of these people also have, muscle issues or they lose muscle mass with their weight. And so that's another thing we're thinking about down the road is whether you could take, a shake like this, which has biological function to, take a lower GLP-1, but and also get the benefits of maintaining muscle strength and getting the same GLP-1 benefits as well, and do it more in a natural way rather than through a fully biotech, driven approach. So
I like that
all of these
thinking.
ideas, yeah,
Mhm.
it's
Nice.
exciting.
All right.
It's a
Yeah.
lot of exciting ideas we have in the future for this axis, this of molecules.
Nice. All right. Well, you've come far, but I'm looking forward to where you head next. Dr. Weiner, it was a pleasure having you on the show.
Yeah. Thanks for having me, Michael. It's good to talk, and I'm looking forward to working with you to help us with some of this clinical stuff in the future.
Fantastic. I'm looking forward to it as well. Thank you.